Hello everyone, and so far in the formal lectures we have been looking at the theoretical aspects as to what really functional genomics mean. So this session is more to introduce how the labs are. So typically what is a lab that carry out some of the assays required for the functional genomics. So this is my lab, and we have group members, close to 12 of them. These include postdoctoral fellows, meaning those who are done PhD, those who are doing PhD, and those who are interested in PhD but having some training in the lab. So what I'll show you is this is a typical lab setup. So we have equipments and instruments that are required for genomic applications, whether to characterize the DNA, RNA or protein, and to quantify them and do some functional assays. Beyond my lab, our department has many other facilities which we routinely use for understanding how the genome functions. So this session is mainly to give you some idea as to what the lab is like, what are the equipments you have, facilities you have, and how these are being used, right? So as you can see here, the lab has several workbenches that supports equipment. So I'll go through some of them. For example, here we have Komal, my student, is working on aspects that regulate how the translational of many of the messengers are regulated in brain. So that is her main project. And right now she is looking at some of the transcript that she is amplifying. You can see that this is typically what you call as a gel box, where you put a gel and you are able to stain the DNA or RNA and look at, and that would give some image as to the quality and quantity of the DNA or the RNA, what you are looking at. Why don't you explain what you are doing?
So I have set the digestion of this particular construct, pcDNA vector, with 2 restriction enzymes, and I've cut it and I've loaded it on gel, and I'm trying to look whether digestion of this particular vector has happened or not.
So basically this is an instrument which has a UV box, which so transilluminator, what you call. So when you stain the DNA RNA with certain stain, for example ethidium bromide or some other such dye, when the DNA or RNA bound dye would excite fluoresce in a different wavelength. So you'll be able to see them easily where the DNA and RNA is. And what I'll show you is some of those images, like you can see here. So these are the images which clearly talks about the DNA. For example, you have 2 bands. This is your size standards. With this you are able to characterize the DNA. But apart from that, we have a couple of other machines. Again, I would request Komal to explain. This is a machine which most of you would have studied in the theoretical lessons that we had discussion. This is a PCR machine, the machine that does the polymerase chain reaction. But it has some difference. But this also can do what is called a real-time PCR, meaning as the products are getting amplified, you can in real time, the same time, you'll be able to measure the changes in the the amount of DNA that is being amplified, right? So this is very sensitive and being used when you are quantifying the difference in the transcript level, for example, right? So that's where it's applicable. Again, I would ask Komal to explain. So this is a reaction going on. Komal, you can explain what it is. Yeah.
So I'll show you with some of my results. So this is one of the results of my experiment, in which I have checked the targets of particular gene. I've tried to look into the transcript level of these targets under a particular condition, in diseased model and wild type, and I'm trying to look at the level of these targets in these diseased models and wild type. These are the CT values, and these shows there is no change in level of these targets. This is how this is how entire plate looks like. We put the plate here for checking the level of these targets, and then after while the reaction is going on, in real time, transcript level of this is recorded, and this is the result that shows that how the level of my targets changes in case of diseased and wild type model.
Right. So that's one application. Likewise, you have again a high throughput machine. This machine has plate. This called as a micro plate reader, which has a 96. Can you open this thing? So it has a 96 wells. Like for example, you can have test tubes or small tubes that are 96. You can put it inside, and then you can do all sorts of assays. For example, it can be used for fluorescence. You can read out the fluorescence. For example, you can grow cells that have a GFP construct under a target, so you can see whether the target is amplified or the target is being turned on in certain conditions, so that can be read out. Or you can have luminescence, for example, or you can have even absorbers. So typically these are the plates that we use. These are, you can see that there are 96 wells. So basically you can do reactions, enzyme kinetics you can culture the cells, and you can put there for reading. So this is how you really put it inside, and then this machine will take it and you can set the temperature. You can enter everything in the computer, and it will give you the results. So it can do absorbance like spectrophotometer that you would have read. Then you can do a fluorimetric reading, and then you can do even luminescence. These are the three things that you can do. There are many such machines. Some of them we'll explain here. So this is a typical lab, molecular biology lab. So we have lab benches, and you can see Rohit we're setting up a reaction, and you can see that you have tubes that are kept over a block. This block is at 4 degrees. So you're setting up a reaction for what you call as a polymerase chain reaction. So you have all the samples and buffers, enzymes kept on the ice, and that is how you maintain, because you don't want to give any thermal shock to the protein, because the protein would become ineffective if you expose them to high temperature. That's its say. And if you come here, this is Kirti, and what she is doing is she's setting up a gel. So why don't you show this? Right, yeah. So this is an apparatus wherein you can cast gel for what you call as polyacrylamide gel. So these are very thin gels which you can use to separate, for example, protein or oligonucleotides or DNA, RNA that are very, very small in size, in bases, 150 bases, 100 bases, and so on. So that's what she's setting up. This is polyacrylamide gel, and these are micro centrifuges. You can use samples for either purification of DNA, RNA, or for concentration of the protein, and you have this. So these are regular machines, what you call as polymerase chain reaction machines, where I'll show you one of them. So you can see here again you have a plate in which you can keep 96 tubes. So therefore you can do reactions of 96 samples. So you can set up the program such that typically in a PCR you have 3 stages, what you call annealing, extension, and denaturation. So you can set up as to what should be the temperature at which you would expect the primer to anneal, extension, and denaturation, and you give a duration depending on what is the size of the product that will be amplified. So that's the typical lab setup. So we have Varsha, an intern, and you can see she's trying to set up gel electrophoresis.
Yeah, I have already set the gel.
Right, so this is...
It is already casted. Now I am going to run my samples. I have already completed my RNA isolation. So I am going to check the quality as well as the concentration.
So this is a typical cast. So what you have is a mold in which you keep the comb. That's where the well kind structure where you can load the DNA, and you melt agarose and leave it there. It sort of makes the gel, and that can be kept inside this apparatus. This is electrophoretic chamber. So you have a buffer and then keep the gel inside and apply the DNA, and then you put them under electric field. Since the DNA is negatively charged, it will move towards positive side, and then you have a gel in between that would allow the smaller DNA to migrate faster. That's how you separate the DNA. Right? So you have another facility here. We can see this is a fluorescence microscope, which is normally used to look at compartments within the cell to see how and what kind of signals come in. This is Saloni.
Right now I am imaging for the HeLa cells, where I am trying to look for the mitochondria, where I am using the 3 filters, FITC, TRITC, and for the blue filters, and I have taken the few images here.
So you can see here, the one that you see in blue color are the one that are identifying the nucleus of the cell, and then you have red color, that is the mitochondria, and the green ones are the cellular cytoplasmic compartment.
Right.
So this is how you look at the cells. We'll go and see how the other tissues are analyzed. For example, if you want to understand function of where is Komal? If you want to understand the function of any gene, so in the reverse genetics, if you remember, we discussed, you delete the gene in the genome and then look at as to what is the function of it in the cell. That's what we have looked at it. So in the microscope, you can look at the cell as to what goes wrong. But you can use transgenic or knockout technology to generate animals that lack a particular gene and try to ask as to what happens at the tissue and organism level. So here Priyanka is looking at one such aspect, that is to look at what goes wrong at the tissue level. So this is, she is basically fixed the tissues of from an animal in which a particular gene has been knocked out, and she is trying to understand what goes wrong at the tissue level. So why don't you explain, Priyanka?
So this is the wax station that we have. We for histological analysis, we embed the tissue in this tissue embedding center, as you can see here. That the tissue, as you know, is very soft. So we need it to embed in a system where we can easily process them for cutting out the thin sections. Like as you know, that we need 5 to 10 micrometer of sections for seeing the histological changes in them. So what we do is, this is the wax station, and in this we embed the tissue in the wax, and then this is the cooling plate where we cool the sections. So where we cool the blocks, and then after the blocks are cooled enough, then this is the cutting section, tissue cutting section. Here, as you can see, we can set the parameters like how much thin sections you want or how much thick sections you want. So according to our need, like right now I need sections that are thin enough, that is around 10 micrometers. So we cut sections of around 10 micrometer, and then as you can see in here, that these are the thin ribbons of tissues, in which, as you can see, this is the brain tissue that I am processing over here. In the block, the brain is over here, which is embedded in this wax, and then this is the 10 micrometer thin section of tissue, which we have to float in warm water, which is around 40 to 42 degrees Celsius, so that the tissue is totally spread out, and then we just carry it out in the slide, just like this. And then we have to dry it overnight at 37 degrees Celsius, so that the tissue spreads out more clearly.
So this is an exclusive facility, what you call as culture room. So all the mammalian cells, human cells or mouse cells, when you do culture for cell biology applications, are done here. So I'm going to take you through this facility and explain briefly what is there. As you can see, it has partitions. This is mainly to prevent any contamination, because such cell culture facilities have the risk of attracting microbes and fungi getting contaminated. So here is the facility. So as you can see here, you have a room which is secluded, and you have what you call as a biosafety cabinet. This is class 2 type cabinet, wherein the air that is thrown into the cabinet is sterilized, and then again it gets into and gets filtered. So you can see you have a flame, you have all the utensils required for the culture, and here is Bhupender who is setting up a culture. Why don't you explain, Bhupender? You can sit and explain.
Okay, so as I told, this is the culture room and we culture cells here. So right now what I'm doing is I have a dish that is full of the cells and the nutrients have been consumed up. So right now I'm transferring some of the cells to the new cells in which new media has been placed so that they can grow in there. And then we can do experiments with these cell lines. These are immortalized or the cancerous cell lines. They have the property to grow indefinitely unless they run short of the nutrients. And this is
Yeah. So we'll show
So we
some
have
of
cells
this.
here.
Yeah.
This one.
So if you have any other stuff?
no, we have
Okay. So these are the what you call as incubators. So basically these are the machines that maintain the temperature at 37 degrees. Not only that, it also maintains the carbon dioxide. Okay, that is to make the medium pH maintained. So I'm just showing you opening one of that to show you that how the incubator looks like, and you can see that it has several different types of dishes. You know, you have a smaller one, bigger one, and what you call a T20 flask. Each one has medium, and cells are growing. Various different kinds of cells are grown within that. Use of the cell line is to understand the function of the genes at the cell level. For example, gene could be regulating a process. The process could be secretion, or gene could be regulating a particular process, say mitochondrial fission, meaning mitochondria getting fragmented, or fusing is one such event, that, or trafficking. You know, there are what you call as endocytosis or exocytosis. There are vesicles that fuse to give a certain function and then to secrete molecules from the cell from inside to outside. So these are the things that you can study using the cells. And when you want to understand the function of certain genes, what you do is you knock down the gene using the RNA approach. We discussed that in our theory class. So in that way, you are able to knock down certain genes and then look at the function of these genes. So this is the typical cell culture facility. Now we are going to look into how the typical cloning and other such kind of molecular biology is being done. So one of the essential component for such work is the freezers. So you can see here, this is an equipment that maintains -20 inside. So you can see already these are ice formed. So you have, for example, all the samples that are stored here. It could be enzymes, it could be DNA, or it could even be some of them the fine chemicals that are required for any of the experiments. And you could see here Komal again working using class 1 laminar flow, in which the air is taken in and you have a filter that sterilizes the air and throws it out via this area. Therefore the chamber within is clean, and she is doing an experiment wherein she is trying to clone a gene, and what you see, the roundish ones are the bacterial dishes. And Komal, why don't you explain?
Yeah, this is ligation mix in which I have cloned, in which I have put my vector along with construct, and I've put ligase enzyme, and I'm going to plate it on plate which has ampicillin that will select the colonies that are specific for my that are specific for my clone. So what I'm going to do is to these are sterilized spatula that I'll use to plate my colonies on this plate. So once the colonies are evenly spread, I'll just seal the plate and will keep the plate in incubator at 37 degrees Celsius for next 12 hours and look for the colonies that are positive for ampicillin and my clone. So likewise, I'll plate all the colonies, I'll plate all the plates likewise, and I'll put them in incubator.
Here is an equipment. This is must have seen in the your labs. These are what is called as centrifuge, but it has more advanced features. You can program the machine such that at what speed you want the rotor to spin, and what should be the speed, how long it should spin, and what should be the speed at which it should come to an halt. And you can also maintain the temperature of the this chamber here. It can go up to sub-zero. You can maintain here 4 degrees. Routinely we do for many other applications. And you can see these are the tubes in which you can either use bacterial cells to pellet them or to purify DNA or RNA. And this is a typical equipment that we use for purifying most of these biomolecules. So what we are going to show you is a storage device. So all the bacterial cells or the mammalian cell lines, or even mouse embryos can be stored for several years without losing their viability. So this is a possibility, and that is done using a technique called cryopreservation. So you mix the cells, either E. coli, bacterial yeast, or mammalian cells, with certain medium, which is, for example, DMSO or the anti-freezing mediums come in, and then you mix them and then store it in liquid nitrogen. So, Rohit, why don't you open it and show? So you can see it's a container which has
two cells are inside. That's here you can see.
So you can see that.
Just be careful.
So you have boxes inside, and then there are tubes that are kept inside, and these are in freezing condition, and normally minus 140, 120. That's the degree Celsius. That's where it's being maintained, and you can revive them anytime that you want. So when you are not using a particular line, this is how it is stored, and later you can use it, revive them using certain protocols. Again, you can culture them and have the cells in running condition. So that's the best way to store. This is another storage device, and you can see here it shows this chamber is maintained at minus 80, right? So the entire chamber inside temperature is minus 80. You can see that. So again, this is another device for storing samples. Anupama, why don't you explain?
Yeah, these are the different racks, and we have boxed our samples whichever are required to be stored at minus 80 in these boxes. So we can keep them safe for years on this temperature. These are the important palatable samples.
So it could be enzymes, it could be proteins, it could be again E. coli, or cells can be stored in this way. So here you have another facility. This is also a culture facility, right? So you have likewise incubators, likewise what you have seen a cell culture facility, where you have incubators, and then you have a very high-end microscope setup. So this this setup is such that you can even image cells that are growing. So basically you have a microscope, and you can put a chamber here. These are the chambers in which you can culture the cells, and you have all these parameters that you can feed in, wherein you maintain the temperature of the chamber, maintain the carbon dioxide level, maintain oxygen level, and then look at the cells when they are growing. So you can look at dynamic changes that are happening within this, and you can use different fluorescence molecules to tag various compartments of the cell, and then you look at how these compartments functions. So to help in setting up the cells for culture and others, we have the biosafety cabinets and other facilities, and we have with us Anshika and Rashmi, who are looking at various aspects. For example, Anshika is looking at some of the non-coding RNAs. We discussed about non-coding RNAs, and some of these non-coding RNAs in the human is expressed only when the cells are under stress. For example, the cells are exposed to very high temperature, say 42 degrees, 45 degrees. So it is a stress on the cell, and the cell has to sort of mount a response, therefore it can survive that high temperature. She characterized one such RNA which is expressed only when the cells are under stress. That is her work, and she would explain some of them here. And then we have Rashmi, and she looks at a different aspect of this, for example, heat shock response, which is when this cell is exposed to very high temperature. What it does is that it also stops the translation process for a while. Therefore, you don't make abnormal proteins. So she's looking into that aspects as to how the translation is halted when the cells are under a stress, right? So what both of them are looking at is to what are the genes that are involved in this process, and therefore we can understand how the process works. And you can see this image here, and you have different colors, and these are nothing but you can see that the blue ones are the nucleus, and you have the green ones. The green ones are the structures called as stress granules. When the cells are under stress, what it does is it puts all the ribosome which are translating the RNA into a compartment and halt their translation. Therefore, the cells do not make protein when they are under stress. And you can explain as to what kind of how these equipments can be used to understand these functions. Maybe start with Ashika.
So, hello. I have been working to study the role of a non-coding RNA in stress response, and I have tried to work on the function of this RNA, that what function it is playing in the cell. And to study that, I have been using some techniques in which I can visualize this RNA, this non-coding RNA in the cell. One of that technique is RNA in situ hybridization. So what we do in that technique is that we use a probe which binds complementarily to the RNA, the sense transcript of the RNA present in the cell, and hence and that probe is tagged to a fluorophore. And when it binds to the its complementary RNA, and we fluoresce the cells in a fluorescent microscope, then that RNA can be visualized. This can be what I just told can be seen in this image. This is an RNA in situ hybridization image, in which the blue color you see is the DAPI, and the pink granules that you see are the RNA which are being probed by an antisense oligo against this RNA, which is tagged to a fluorophore. So this is called fluorescence in situ hybridization. And this microscope that you see here, it is a fluorescence microscope. That's the basic principle of this microscope, and it emits as we can expose the cells to different wavelengths, and according to the fluorophore tagged to the proteins or nucleic acids that we want to visualize, and the wavelength of light that we emit, accordingly we see a colored signal in the So that's the basic principle of the microscopy, the fluorescence microscopy. And Rashmi will tell you more about her work.
Okay, so as I already told you that cells, when exposed to different stresses, then they respond to maintain homeostasis. So they form stress granule by halting translation, and that is here this green you can see, these are the stress granules. This is formed when when translation is halted, and this normally get whenever this cells are getting recovered, they get cleared via autophagy or so you can see here I am just looking on this aspect, and this pink granules you can see, these are the autophagic marker, that P62. And apart from this, from this microscope, we can do live cell imaging as well. And here is that experiment was done. And here this gray, it's a DIC image, and this green, this is overexpression construct, GFP tagged. This is Laforen LDH GFP, and this red one, It is RFP-TR1. It is another stress granule marker, and the cells were exposed to heat shock. So for this I have used this live cell imaging whole facility, and for this we can use, this is a chamber where we keep 35 mm glass bottom dish inside this, and then we maintain temperature and CO2 via these modules and humidity as well by setting all the parameters, and then we can maintain all the parameters, and we visualize what happening in the inside the cell. Apart from this live cell imaging, we have two other microscopes as well. Here one is inverted one, and another one is stereo microscope, or you can say it's a dissecting microscope. So this normally we use to dissect out minute brain, dissecting brain from the mice embryo, and this we use to visualize cells morphology.